Hi everyone, we're happy to have you watch this video from a series of talks called Light or Lite in Swedish. The series of presentations is a joint effort between two of SciLife Lab's research infrastructures, the National Genomics Infrastructure or NGI, and the National Bioinformatics Infrastructure of Sweden, which is popularly known as NBIS. My name is Renuka Kudva, and I'm a project coordinator at NGI at SciLife Lab. And it is my pleasure to introduce you to the contents of this presentation by Richelle Björvang, a data steward at NBIS, who will cover what to consider while working with human data. So Richelle will take us through definitions and dive into how to ensure that one is working within stipulated frameworks for sensitive data. Now, while this presentation is indeed very detailed, it serves to be general, and you are encouraged to contact us if you have specific questions about your research project, as we're always happy to help. And all the links that Richelle mentions during her talk can be found in the description field for this video. So what are we waiting for? Let's listen to what Richelle has to say.
Hello, everyone, and welcome to this presentation on research involving human data. What do I need to think of as a researcher? I am Richelle Björvang, and I am a data steward at the NBIS Data Management Team. This video is a joint collaboration between NGI and NBIS. So for today's agenda, we will talk about what personal data is and when does it become sensitive. What do I need to think of when doing research involving human data? Some examples of secure management of personal data, and how can I share research data collected from human participants? So that we are all on the same page, I will begin with some definitions of terms. So human data is any data associated with or derived from a human being. But when that information refers to an identifiable living person, it is already counted as a personal data. There are certain special categories of personal data that need extra protection, which we call sensitive personal data. When the personal data has information that is used to identify an individual is replaced with a pseudonym or code, then that is pseudonymized data. In pseudonymized data, the code key is locked away and accessed only by certain people. But when this code key is destroyed, it is then called an anonymized data because it is personal data that has been stripped off of any personally identifiable information. So what are the special categories of sensitive personal data? So here, as defined by the GDPR or General Data Protection Regulation, this is information that refers to information that are particularly private or sensitive in nature. So this special category of personal data includes health data, genetic data, sex life or sexual orientation, trade union membership, racial or ethnic origin, religious or philosophical beliefs, biometric data, and political opinions. These types of data require extra protection due to its potential for harm or discrimination if mishandled or disclosed without consent. So now that we have our definitions in order and we're all on the same page, I'll talk about how to deal with human data in a research project. And here you have three different aspects. So we have planning, execution, and conclusion. For planning, here you draft a data management plan or a DMP, and then you register your personal data processing operations. You make a risk analysis, make a data protection impact assessment or DPIA if necessary, and you have to have ethical approval. For the execution, you collect informed consent if necessary. You document your decisions regarding the processing of the personal data, although this should have been done much more before processing even begins. And then you also follow good information security practices. And once you have finished your project, you share data for secondary use, either delete or archive the data, or also archive the project documentation. So I will go through these different aspects one by one. So we start with planning. drafting a data management plan or DMP. So these are some of the questions that we ask when drafting a data management plan. What data will be collected or used? How will the data be organized? Or how will the data be documented? And how will the data be preserved and made available for secondary use? So there are actually several useful tools online. So we have this DMP online, which is offered by most Swedish universities, but is not specific for life science. SciLife Lab has have this data stewardship wizard that is really good and is actually developed for Swedish life science research. Once you have the DMP, you register your personal data processing operations. And these are the questions that are answered in doing that. So what is the purpose of the data processing? What will be the lawful ground for data processing? What personal data will be processed? Will the personal data contain any sensitive data? How will the data be stored and processed, and which parts of the information are to be deleted or saved when the project is finished? After this, you will then make a risk analysis of your personal data. So you analyze the risk and suggest appropriate information security measures about confidentiality, integrity, and accessibility. Confidentiality is about measures to ensure that the data is kept confidential from those that do not have rights to access the data. Integrity is about measures to ensure that the data is not corrupted or destroyed. And accessibility is about measures to ensure that data can be accessed by those who have the right to access it when they need to access it. So it is quite important to establish adequate information security measures, and this should always be done for all types of research data, but it's even more important for human data. And the information security measures are both procedural and technical. And the information security measures that need to be established should be defined at the planning stage when doing a risk assessment so that you'll be able to know if there are any impact assessment that needs to be done. So here, you make a data protection impact assessment if your processing is likely to result in a high risk to individuals, people's rights, and freedom. So you use your risk analysis, the step before this, to decide if you need to do a DPIA. If your risk analysis results to a high risk, you make the DPIA. We need to know also that not all risks can be eliminated, but the DPIA can allow you to identify and mitigate against data protection risks. So plan for the implementation of any solutions to those risks and be able to assess the viability of the project at an early stage. So if you're in doubt if you need to make a DPIA, it's the safest ways to just make a DPIA. There are different tools that exist, and your data controller, which is usually your university, can probably give you some guidance on how to do it. It is also important then that you receive ethical approval from the Swedish Ethical Review Authority before you begin your actual research. So when you're applying for this approval, you'll be asked about the data handling of the project. So for the execution, it's very important to have informed consent. If your research project requires it, each research participant should be given information about the data processing before he or she decides to participate in research. So it's important to note that there are different aspects that are required based on, let's say, Ethical Review Act, and also based on GDPR or General Data Protection Regulation. So make sure that all this information that are listed here are included in the informed consent so that your participant is fully informed before he or she decides to participate. So we talked about pseudonymized personal data and how to use pseudonymized data. So first, know what is the identify first the personally identifiable information. So let's say this is the personal number. And then you generate pseudonyms. So the pseudonyms are unique identifiers to replace the original personal identifiers. So For example, this could be ABC123 or something to this pattern. And then you replace the PII with these pseudonyms. Then you document the mapping, meaning you keep a record of the mapping between the original identifiers and the pseudonyms, since this is very crucial for any future reference or data analysis. It's also important that you secure the storage of this data to ensure that both the original dataset and the pseudonymized dataset are stored securely to prevent unauthorized access. So it's important to limit the access, to control the access only to authorized personnel to work with it. And important, of course, to adhere to regulations to make sure that your pseudonymized process complies with relevant data protection regulations such as GDPR. Last step is to validate the pseudonymized data to ensure that it still maintains the integrity and is usable for research purposes. Regarding encryption of your data, so your encryption transforms data into an unreadable format, protecting it from unauthorized access. There are strong algorithms that ensures that even if data is intercepted, it remains unreadable. And with this strong, good practice is to encrypt your data at rest, for instance, the raw data when it's not in use. And if you want to try out encryption after this presentation and see how it works, for those with Mac or Windows, you can try the 7-zip on your local computer, and it follows the SHA 256 encryption. So it's a good encryption program to use. It's also important to back up your data. So there is a backup strategy called the 3-2-1 backup principle, where you have 3 copies of the data. 2 of those copies are in different media, and 1 copy is in different geographical location. So don't forget to test your backup solution and remember to document your backup strategies as well. The third concept of secure data management regards the restriction of access to the data. So does everyone have in the project need access to all the data all the time? So it's important to assign permissions based on individuals' roles and responsibility within the research project. And make sure to use strong passwords or password manager, or even use two-factor authentication or multi-factor authentication to be able to make sure that the data has restricted access. Once you have finished data collection, is about to conclude your project, here are some options for sharing your personal data for secondary use. So either you publish it in a research paper, but make sure that there is no personal data in the paper, it's just the findings or aggregated results. You can also use public repository for sharing anonymized data. So for example, here is Array Express as a repository, because this has, for example, aggregated data or RNA sequence count matrices. Then we have the controlled access repositories for sharing sensitive data. And in Sweden, we're happy to present FEGA Sweden, which is Federated European Genome Phenome Archive Sweden, which is a national repository for sharing sensitive personal data from Swedish life science research. Also, it is important to note that the research data should be stored for at least 10 years. But personal data that is not needed to reproduce results should be deleted. And document the necessity of retaining any personal data and keep it safe and secure. So in summary, planning is key to a successful research project. Make sure to plan your personal data processing before you start processing the data. So draft a data management plan, register your personal data processing operations, and make a risk analysis and a DPIA if necessary. It's also important to follow good information security practices throughout your project, so you encrypt files, have a backup strategy, and restrict access, and share anonymized data publicly, but share personal data under controlled access. So open as possible, and as close as necessary. If you want to read more about this, we have the SciLife Lab RDM guidelines, which is a knowledge hub for the management of life science research data in Sweden. We also have the RDM Kit, a research data management toolkit for life sciences. And you have FEGA Sweden, which is a national repository for sharing sensitive personal data from Swedish life science research. If you have any more questions, please feel free to contact your data protection officer at your university or your university's research data support. Or you can also email us at SciLife Lab, the SciLife Lab Research Data Management Support, which is data-management at scilifelab.se. Or you can also email the FEGA Sweden help desk at ega-se@nbis.se. Thank you so much for listening to this presentation.